Let's talk about the brain. So we're going to work our way up through the structure of the brain, starting at some of these oldest areas, down here in the spinal cord and just moving up in through the brain stem, up through the midbrain, and then we'll add on the cerebrum, which will be out on top of this. As we go, we'll try to keep track of what these different areas are doing. So to start with, we've got the spinal cord coming up here, and at the top of the spinal cord is an area roughly here called the medulla oblongata. Evolutionarily speaking, this is the most primitive part of the brain. It's the oldest, it's the furthest back down on the neural tube. In that previous diagram where we talked about those three initial vesicles, the rhombencephalon, the mesencephalon, and the prosencephalon, the rhombencephalon gives rise to this area. This is the lowest down area. And the medulla oblongata is involved in regulating some very basic autonomic functions. So some things like blood pressure and regulating breathing. The medulla kind of keeps track of some of those basic homeostatic functions and handles those, which makes sense given its age. Pretty much everything that has a brain that's organized, anything at all like this brain, has a medulla oblongata. And those are functions that are common for most organisms. Now, later on, when we talk in the peripheral nervous system, we're going to talk about these sympathetic and parasympathetic branches and how those often produce sort of opposite effects in the body. The medulla plays a big role in directing both of those areas, the parasympathetic and sympathetic. So, kind of falling under that idea of basic autonomic function regulation. It's also one of the reasons why damage to this area is so devastating. Strokes that affect the medulla are often fatal. Just because if you mess this up, you've messed up stuff that handles the basic functions of keeping the body going. So, hopefully that makes sense. Coming up a little bit from that, we have these two. We have this area, which is the pons, and this area right behind it, which is the cerebellum. These two together also come from that rhombencephalon. That original rhombencephalon divides up into the myelencephalon and the metencephalon. And the metencephalon becomes the pons and the cerebellum. And we're going to start with the pons. So the pons, the word means bridge, and that's largely what this is. It's actually the gatekeeper to the cerebellum. So one of the biggest functions of the pons is to regulate the flow of information to and from the cerebellum. So this regulates communication with the cerebellum. It also works with the medulla in regulating a lot of basic autonomic functions. So for example, regulating automatic breathing is handled between these two areas, between the medulla and the pons. They both play a role in that. The pons also has one other particularly interesting job, which is that it plays a role in sleep and dreaming. Now, dreaming is interesting. At this point, we still don't know why we dream. We still don't know why dreaming is so important to the human nervous system. We do know that I think all mammals, and I believe most animals, do dream as far as we can tell. But we still don't know exactly what it's for. We do know it's necessary. If you take, for example, rats and disrupt their sleep every time it looks like they start dreaming, eventually they die. We don't do that experiment in humans, but if you do sleep deprivation experiments in humans, if you prevent people from dreaming, it does mess with their minds, and it causes relatively serious issues with their bodies. So we know it's important. Now, when you're dreaming, at least during the active dreaming sleep, the activity in your cerebral cortex is fairly similar to what it's like when you're awake, which is probably why the experience of dreaming is sort of like a weird version of actual conscious experience. But there's an interesting phenomenon that can happen. You may have had this happen to you. It happens especially often if you wake up out of a bad dream, like a nightmare. You wake up suddenly and you can't move. This is this phenomenon of sleep paralysis, where you come out of a dream and you are unable to move. Often you're unable even to feel like you can take a really deep breath, and you can't speak. It can be very frightening if you come up out of a nightmare this way, where you're having an unpleasant dream and you wake up and you can't move and you can't really breathe, and you can't yell, and it can be scary. It happens because of something that happens here in the pons. When you're dreaming, your cortex is behaving very much as it does when you're awake, which means it is trying to send commands to your body to do the things that you're doing in your dream. If you dream that you're running, your cerebral cortex is trying to send commands to your body to make you move as if you were running. The pons seems to be the area that stops that. It puts on a block that prevents you from sending commands out to your body to do it. It kind of cuts off your brain from your body. Your body stays alive, obviously, and you keep breathing, but it prevents you from directing your body the same way, which is generally good. You generally do not want to act out what's going on in your dreams. But, I mean, it's just a safety issue. But when that block is placed and then doesn't get released as soon as you wake up, you end up with this interesting thing where you're awake and you're trying to move your body, but your pons hasn't released that cutoff yet. So when you suddenly wake up, sometimes the pons takes a moment to catch up, and until it does, you're paralyzed, which is an unusual and sometimes unpleasant phenomenon. Anyway, so the pons has some interesting things there. Now the cerebellum, living back here, if you've seen a picture of the brain or seen a brain, you know that there's kind of a big wrinkly bit. It's right here in the back, kind of tucked partway under the cortex. That's the cerebellum. It's wrinkled because there is so much stuff going on in there. It has a very, very large cortex with something like as many cells as the rest of the brain put together. And it does some really interesting things. The major role of the cerebellum is in motor coordination. But not just coordination, it's also in correction. That's kind of an interesting thing. So here's kind of what we're talking about. When you decide, say I decide I'm going to reach out and touch the word cerebellum on there. So my cortex comes up with the sequence of movements I'm going to need to do that. It says you're going to need to move your shoulder this way and extend your arm this way and put your hand about here. So it figures out what movements are going to be necessary, and then it starts issuing those commands. Now, when I decide what I want to do, the cerebellum gets, we're not quite sure how this works, but it seems to get some sort of copy of that, saying, this is the desired movement. And then as we start to do it, our eyes and our proprioceptors, the sensors in my muscles and joints that tell me where my body is, are constantly sending updates and saying, here's what's actually happening. So that as I issue the command, the cerebellum says, here's what I want, here's what I'm getting, and then the cerebellum helps to direct any corrections that are needed. So for example, let's say I start reaching up but I'm off a little bit. My cerebellum detects that and corrects the movement to help me get to where I want to go. When all of that is working properly, we're capable of moving in smooth and coordinated ways, so that our cerebellum is constantly correcting our movement to get what we want rather than what we're actually getting from the commands that my motor cortex issues. Now, people with damage to the cerebellum often have movements that are poorly directed. So a person with cerebellar damage might want to reach out and touch that word, but have to constantly correct it as they go, have to kind of crudely correct it rather than nice, smooth, constantly updated corrections. So the cerebellum is responsible for our ability to move in ways that accomplish our goals efficiently. So every time you walk nicely or reach up and grab something or catch a ball or something like that, you can thank your cerebellum for the nice job it's doing of letting you smoothly coordinate your movements. All right, those three structures that come from that rhombencephalon are sometimes what we refer to as the brain stem. The next area up here, the midbrain, sometimes is considered part of the brainstem and sometimes not. But let's get to that in just a moment. All right, let's get into the midbrain. So that's this area right here. So the midbrain is kind of divided into two areas. There's an upper tectum and the lower tegmentum. the tegmentum is related to the functioning of the pons and the medulla. It's handling a lot of autonomic homeostatic functions. One interesting thing that happens in the tegmentum is that, depending on what source you read, either starting here or starting here, we have this upward going system of connections called the reticular activating system. This reticular activating system seems to have a big role to play in regulating sort of states of consciousness. So the difference between, this may be happening to you right now, for example. You may be sort of sitting there, kind of zoning out, not really fully focused on what's going on. Yeah, your brain's kind of wandering. And then the instructor says, you there! And you say, oh, what? Up, yes, answer, I have an answer. That switch from not fully there to fully engaged, ready, awake, and conscious, and active and processing sensor information seems to be something that the reticular activating system does, regulating those states of consciousness. Now, of course, when from your instructor they're saying you should always be fully engaged and aware and awake, and of course that's not how humans work. Sometimes you need to be disengaged. Honestly, sleep is just one level on that. You go from fully awake and engaged to kind of zoned out to kind of drowsy to unconscious. Those are all normal parts of human consciousness. But the ability to switch between them, what we sometimes call states of arousal, by which I do not mean sexual arousal, but awakeness and engagement, seems to be something the reticular activating system is involved in. So that's kind of an interesting thing. Now the tectum does something of particular interest to me. So the tectum is divided in humans into a couple of specific areas. One area called the, I'll label it here, SC and IC, and that's the superior colliculus and the inferior colliculus. If you've heard of something called the quadrigeminal plate, that's what we're talking about here. Because if you look at it from this direction, it kind of looks like 4 little bumps. Those are the superior colliculi and the inferior colliculi. The inferior colliculi play a big role in our ability to locate and process sound, which is actually an interesting process neurologically. The ability to locate a sound involves processing the difference in input between your 2 ears. And there's several things that go into it. One of them seems to be the timing, that the sound waves arriving on one side of my head and then the other has a slight difference in timing, which, because of some pretty sophisticated neurological connections in here, your brain can analyze. Also, as a sound passes through and over and around your head, it changes what frequencies you hear on one side versus the other. So differences in what the sound sounds like between the 2 ears also affect your ability to tell where the sound is coming from. Put all that together, and when you have a sound over here, I have the distinct sense that it's coming from a location in space. And if you move it, I can locate roughly where it is. There are other animals much better at this than humans. For example, animals that can rotate their ears around can use that to help pin down the location of sound. We can't move our pinnae, although interestingly, the muscles to do it are still there. If you can wiggle your ears, let me see if I can do it. The muscles that you use to wiggle your ears are the vestigial remnants of the same ones that cats use to swivel their ears around. But anyway, so as sound comes in, the inferior colliculus is one of the areas that processes it and helps to locate sounds in space. So there kind of is a map on the surface of the inferior colliculi of space around us, and a sound coming from some particular location will tend to cause activity in cells on that map corresponding to where it is. More on that in a second. The superior colliculus gets information from the inferior, so this sound information is brought forward, but it also gets information from the visual system. And the superior colliculus is involved in eye movements and visual and auditory space. The superior colliculus is pretty interesting. So the eyes are sending information. The optic nerve comes mostly to an area called the thalamus, which would be about here. But they also send a separate feed here to the superior colliculus. And on the surface of the superior colliculus, we have cells that are organized based on both where visual stimuli are in space around us and auditory. And the superior colliculus seems to be involved in sort of keeping those two things together so that we see and hear things coming from consistent locations. So, for example, if there's a bird out here and it goes cheep, cheep, my sound processing at the inferior colliculus says, where's that sound coming from? My visual system says, there's a bird there, and the superior seems to sort of put those together and say, okay, visual space, auditory space, they're all together, they're all calibrated. Which means you can do some interesting things with this. A guy named Knudsen did an experiment where he took baby barn owls. Barn owls, their area here is called the Tectum. They don't exactly have the same kind of colliculi, but it does pretty much the same thing. These barn owls were raised, some of them he glued prisms onto their faces, which shifted their visual world over something like 15 degrees off. So they saw everything not quite where it really was. Now, barn owls use visual and auditory space for navigation. And so in these little barn owls, when he put these prisms on their faces, they couldn't get around. They'd fly into things and bump into things. They had a terrible time of it until they got used to it and somehow adapted to it. And when he investigated how their colliculi were organized, what he found is that right at first when he put the prisms off, visual and auditory space were off. That if you had a visual and auditory source, it would show up at a different spot on the superior colliculus for visual than it did for auditory. But over time, as they adapted, their map of auditory space changed. This area changed how it was wired together to bring the auditory registration in with the visual one. So they shifted their map of auditory space to match visual space, which is interesting for two reasons. One, it means this area is rewiring itself, altering which neurons are connected to which other neurons. And two, they switched the auditory space, which was correct, to match the visual space, which was wrong. So the visual space seemed to be sort of an override, and they remapped auditory space to work with that. Now, even though they were both wrong, they were still able to use this to direct their movements properly. So once they adapted, they were able to use the maps, because they were now all together, to get around properly, which is pretty interesting. And then, of course, when he took the prisms off, now their maps were off again, and they had a terrible time until they readapted. And there seemed to be an age after which they couldn't readapt. That if the prisms were still on at that point, those areas were no longer as able to rewire, and they just had problems forever. Poor little owls. But anyway, now my own work in grad school involved not the same kind of experiment, but it involved taking people and having them wear a set of goggles which flipped their world upside down and backwards. So they saw everything flipped left, right, and up, down. And they had to not see the world without those goggles for 2 weeks. They had to live with those. They had to be blindfolded when they weren't wearing them. We did a lot of experiments on these folks, but one of them was that they'd be sitting here looking straight ahead, and they could only see maybe this much. And what they saw was backwards. Then I would have my hand out here where they couldn't see it and start snapping. I'd say, point to the sound. And they'd point to it. I'd move it forward and they'd keep pointing to it. But the moment it entered where they could see it, so now they're seeing it over here, they'd point to where they saw it. They said, yes, it sounds like it's coming from there. And as I kept moving, they'd point to where they saw it until it got out of their visual field, and then they'd point to where it really was. So as long as they could see it in the wrong place, they felt like they heard it coming from where they saw it, rather than where it was actually coming from. But the moment that they couldn't see it, they heard it correctly. It was a very strange result, very interesting. Anyway, so that's kind of the midbrain. It's involved in a lot of the same sort of regulation of basic autonomic states, regulating arousal levels, and keeping track of visual and auditory space. one last thing about that. If you've ever read or watched Little House on the Prairie, you know that there's a character on there, Mary Ingalls, who went blind at one point. She lost her sight. Now, she's based on a real-life person, Mary. There was a real Mary Ingalls, and she did go blind. The real Mary Ingalls was known to track objects with her eyes, even though she couldn't see them. Probably because she went blind, likely because of damage to her visual cortex, which is here in the back of her head. That's where conscious sight happens. So if that area isn't working, she can't consciously see. But the Superiorcolliculus was still getting information from her eyes, which could be used to direct eye movements. So even though she didn't know she was seeing something, her brain could still track the object to some degree. This is a phenomenon known as blind sight, and you still see it in some people with certain kinds of blindness. Anyway, all right, so that covers this area. We're now done with this, and we're ready to move on. So in that original three-vesicle model, we had the rhombencephalon and the mesencephalon. Those became the myelencephalon and the metencephalon from the rhombencephalon. The mesencephalon becomes the midbrain. Now we had that forward one, the prosencephalon. And when that developed into other vesicles, it becomes the diencephalon, which is going to be the thalamus and hypothalamus, and the telencephalon, which is going to become the cerebrum. So that's the area we're getting to next. Now we're ready for the structures of what we call the diencephalon, which was one of the outputs of that last chamber, the prosencephalon. That became the diencephalon and the telencephalon. So this is the diencephalon. In the mature brain, there are two main structures that come from that. Here in brown, the thalamus, and in orange, the hypothalamus, which we'll get to in a moment. So the thalamus, it's kind of right there in the very middle of your brain. It's a structure that is pretty much, it's one structure that kind of divides partway into two parts. We're going to get to the division to left and right hemispheres, but the thalamus is still at least partially together. And it does several things. Probably its main function, as far as we know right now, is that it's where sensory information goes before it goes out to the cortex. So, for example, the feed from your eyes comes first to your thalamus. The thalamus processes and filters it and then sends it on to the visual cortex, which will be back here. When you hear, sound comes in through the brainstem, processed at the inferior colliculus, heads up to the thalamus, and then out to the auditory cortex. When you feel, that sensory information comes up through here to the thalamus, actually usually to the medulla, then to the thalamus, and then out to the cortex. The cortex is where we have the conscious perception of these sensory things. So this happens before that. And it seems to be involved in relaying that sensory information, processing, and probably filtering it. So processing and filtering sensory information. All of your senses, except smell, come through the thalamus. So all of the conscious senses are processed by the thalamus first, except smell, before they head off to the cortex. Smell is actually the oldest sense, and it goes through a different pathway. But we now think that the thalamus plays a role in filtering and letting us attend to stimuli. So rather than constantly having all sensory stimuli come in at the same weight, your thalamus seems to let you focus on certain stimuli. So for example, right now you're probably mostly experiencing the sound of my voice and not the background noises of wherever you are. Maybe a computer fan running, or a bird song, or the rumble of cars on the freeway. All of those sounds are there, and now that I've mentioned them, you may be hearing them better. But when you're not paying attention to them, it may be that it's the thalamus that filters that out and lets you focus on particular sensory information, which is a useful thing to do. We kind of have to do that. There seems to be sort of a feedback loop between the thalamus and the cortex where we experience that, almost as if the thalamus sends it on to the cortex talks back and says, pay attention to this part, and the thalamus filters other stuff out and sends the new information to the cortex. There are some people who believe that that's actually kind of at the root of what it means to be conscious, our ability to monitor and regulate what we're attending to. So the thalamus does actually seem to play an important role in at least some aspects of what it means to be a conscious individual. Kind of interesting. There's a lot of research on the thalamus right now. Now, equally important, although, well, interestingly and differently important, is this area called the hypothalamus. In terms of human physiology, this is a really fascinating area because the hypothalamus is one of the major interfaces between the body, between the brain, your mind, and body functions. So the hypothalamus, first of all, parts of it act as a gland. If there is a master gland in the body, it's the hypothalamus. Even though it's nerve tissue, it also works like a gland. And it's connected to this thing, the pituitary gland, which also plays a major role in controlling the endocrine system. We'll talk about that when we get to the endocrine later in the class. So this has a big role in controlling endocrine functions. It also is a major regulator of the autonomic nervous system, the sympathetic and parasympathetic, which we're going to get to. And the hypothalamus seems to play a role in how our body processes sort of basic behavioral drives and even sort of basic emotion. So when you feel hungry, that's really your hypothalamus talking. Your hypothalamus is monitoring aspects of things like blood glucose and sending signals to the rest of your brain that says, you want to get food, giving you that basic motivational drive of, I'm going to go get some food, or thirsty, or probably to some degree sleepy. The hypothalamus seems to play a role in regulating circadian rhythms. It is also probably involved in sex drive. Some of those other basic motivations like libido probably live largely in the hypothalamus. The hypothalamus is actually part of kind of a network of areas, some of which are in the next area we're going to talk about, the cerebrum, which we sometimes call the limbic system, that seems to be involved in kind of low-level emotion and automatic reactions. But we'll get to that in a second. So this hypothalamus is the major interface between the nervous and endocrine systems, and it's involved in basic drives and motivations. Okay. Now, that's the one other last thing. I think we saw in the previous diagram this thing about the eye cups that are actually part of the developing neural tube. Those connect here to the thalamus, so they come from the diencephalon. All right, now that we've done that, we now move on and add the part of the brain that you're probably more familiar with as the brain, which we call the cerebrum, that comes from that last vesicle, the telencephalon. So to talk about that, I'm going to erase this stuff. And to draw in the cerebrum, I'm going to have to sort of overlay it on this. So I'm going to draw over the cerebrum and then alter this a little bit. Okay. That probably is a little bit too small given the size of the brainstem we were working with, but it'll do for now. Now, I'm also going to draw this in sort of a different kind of cross-section. That's not a very good drawing, but it'll do for now. Okay. So, let's color in a few of these things here. Okay. So the brain stem, all of that stuff that we've been talking about so far, was all in this lower part. And then from there, that final vesicle, the Diencephalon, if we looked at our neural tube, grew out and split into two big swellings. Those end up being, that final area is called the cerebrum. And it is largely divided into two separate hemispheres. So when we think of our brain, we often think of a left and right hemisphere. All of this other stuff, all of the brainstem, midbrain, and diencephalon is still a unified structure. There's one of those. But then from there, it splits outward into two big, largely separate cerebral hemispheres. Now, those cerebral hemispheres are divided up into gray and white matter, like we talked about last lecture. And there's a lot of deep areas of gray matter that are sort of buried down in the cerebrum. And then there's also an outer layer of gray matter. And I'm sketching in here. So this outer layer is the cerebral cortex, whereas these other deeper areas we usually refer to as various kinds of nuclei. Different from atomic nuclei. Nucleus is just the name for a cluster of cell bodies inside the central nervous system as opposed to out on its surface. So, let's talk a little bit about some of those deep areas first. Now, we're not going to list all of them. They're complicated and there's a lot of them. But some that we might talk about would be things like the amygdala and hippocampus. Those are kind of found deep in this part. The basal nuclei. That's actually a decent list. That's not all of them by any means, but that's some of the ones we're going to mention. These areas, let's talk a little bit about what these do. The amygdala, which are kind of deep in this part, along here, there is a deep area of gray matter in the brain that seems to be involved largely in fear and aversion. We know that the amygdala, activity in the amygdala produces feelings of aversion or fear of things. And we also know that the amygdala is part of how we judge, or actually it's maybe part of the judgment of my group versus others. There's some really interesting research out there that people develop a sense of who is part of my group and who is part of everyone else. And when you're exposed to people who your brain classifies as everyone else, the amygdala has some level of response. That doesn't mean people are inherently terrified of everyone who isn't part of their family. But it does seem to mean that we have some level of deep-set pre-wiring for my clan and not my clan. And not my clan is a potential danger and my clan is trusted. This has some obvious implications for how humans live together. And there's some really interesting research out there that you can actually participate in. If you haven't done it, you might look into the concept of implicit bias and even look up the implicit bias test. If you get a chance, it might be worth taking. The implicit bias tests investigate to find out what kind of automatic level associations you have between faces of different appearances and words, for example, although there are other things, with different emotional weightings. And this can be very insightful because all of us grow up with this whole amygdalar-based thing of my group and the out group, and we tend to associate more positive emotions with my group and more negative ones with the out group. But who we place in those groups and how we classify them is very interesting. The implicit bias tests can help you identify whether or not you have any sort of conscious biases. They can help you identify where you may have unconscious biases that you don't even know about. Some level of deep-seated automatic association of faces that have a certain look and words that have a certain valence. It's inevitable that people have these. We grow up in, this is part of the way our brain is wired, and we grow up in a culture that encourages it. But if you don't want to have those biases, finding out that they're there is the first step to seeing if you can find a way to work around them or to mitigate them. So it's something you might consider doing. It's a very interesting thing, and you may learn some interesting things about yourself that can then guide your future behavior. I recommend it. Anyway, so the hippocampus, which is very close to and connected to the amygdala, is involved in several things. Among them are probably spatial navigation and memory encoding. We don't know how long-term memory works in the brain at this point, but we do know that the hippocampus seems to be involved in taking short-term memories and encoding them in some way into long-term memories. We don't know how it works, but we do know that it happens there. And you may have seen a movie called Memento, which was about a fictional person who couldn't form new memories and how they had to deal with a crisis. It's based loosely on a real person, probably the most famous neurology patient ever, patient HM, who died not that long ago, who had both of their temporal lobes removed to help with epilepsy, and ended up with the inability to form new memories. Now, they could learn new skills, interestingly, but they could not form new conscious, what we call declarative memories, which meant that they always thought that they had first woken up after their surgery about 5 minutes ago. Even decades later, they thought that they were newly awake, which was fascinating and possibly kind of frightening, really. Anyway, interesting case. So, the basal nuclei down in here are involved in, they're involved a lot in some levels of motor control and coordination. They kind of work with the motor cortex and the cerebellum. Now, we mentioned that the hypothalamus was part of a network that handled some low-level emotional and behavioral issues. The amygdala and hippocampus are also part of that system, which altogether we sometimes call the limbic system, which is kind of fun. Some people talk about the limbic system as being part of the lizard brain, the idea being that it feels kind of primitive. If you know anything about Freudian psychoanalysis, which I know is somewhat outdated, but there are some interesting concepts in it still. The idea of the id, this part of our mind that is primarily focused with satisfying our desires. This kind of feels like the id. The limbic system seems to be responsible for kind of basic emotions and gut reactions. So if I'm driving along the freeway and someone cuts me off, I may feel, kill, kill! That kind of instinctive rage, anger, fear, or even some levels of lust or love. This thing, I want that. Those all seem to be limbic system functions. Some of these higher areas of the brain seem to be involved in telling the limbic system no. We'll get to that in a second when we talk about the frontal lobe. But the limbic system is also seems to be involved in the areas that are responsible for pleasure and satisfaction and addiction. So the fact that you do something you enjoy doing, you eat your favorite food, or hear a sound, I don't know, but you do something that you enjoy, that feels satisfying. That satisfaction comes from the limbic system. You're satisfying that basic need seems to be a limbic system function, and it feels great. The problem there where you say, okay, and now I want that all the time, that level of addiction, I can't feel happy without that. That also seems to be a limbic system function. So pleasure, reward, and addiction also fall under the limbic system. So keep that in mind, this hypothalamus, amygdala, hippocampus, this network of interconnected areas that seem to be involved in satisfaction, basic emotions, and drives. Alrighty. So now moving up from these deep areas, we're now going to move out into this layer of gray matter out here on the outside, the cerebral cortex. And I'm going to talk about that in the next part of this lecture.